Aflatoxin B1 Rapid Test Kit Operation Video. The operator should wear laboratory coat, mask, and gloves. 1. Preparations. Allow test strips, microwells reagents and sample diluting buffer reach to room temperature before use. Mix 500 ml ethanol and 500 ml deionized water well. Get 50% ethanol water solution and ready to use. 2. Sample pretreatment. Mix the tested sample well. Grind the sample about 3 to 4 times. Pass the ground sample through a 20-mesh screen. Weigh out 5 grams of ground sample into a 50 ml centrifuge tube. Add 12 ml of 50% ethanol water solution to the 50 ml centrifuge tube, which contains the sample. Tighten the cover and shake by hand, then vortex fully for 3 minutes. Transfer the mixed sample to 2 2 ml centrifuge tubes. Centrifuge for 5 minutes at 4,000 revolution per minute and get the top layer of extract. According to the cutoffs in the table, add sample diluting buffer to the tube. This video demonstrates with the cutoff of 5 parts per billion. Vortex and mix the diluted sample well. That is the sample to be tested. 3. Test procedure. Take out the required microwells. Place them in the microwells holder and mark the test strip. Add 150 microliters of sample solution to each gold microwell and pipette up and down for 5 to 6 times until the gold standard in the microwell is completely dissolved. Incubate for 3 minutes at room temperature. After the incubation, put 1 test strip into 1 well immediately to make the handle with logo upward and the other end downward and fully immerse into the solution. Incubates for 8 minutes at room temperature. Take out the strip and remove the absorbing pad immediately. Interpret the test results according to the image within 1 minute. Other time interpretation is invalid.